Good evening to the class. Today we're going to discuss about endomembrane system. Right. First of all, let me tell you about a small thing. We have our body is made up of different types of cells, and each cell is having different cell organelles inside it. If you want to name some of the cell organelles, you will start with mitochondria, chloroplast, endoplasmic reticulum, lysosomes, spirosomes, vacuoles, etc., etc., etc. But among these cell organelles, there are especially 4 cell organelles, only 4, which act coordinatedly. How do they act? These endomembrane system organelles, they act coordinatedly. They act coordinatedly, and this endomembrane system is specifically seen only in eukaryotes. Seen only in karyotes. U means true and karyotes means nucleus, which means like in human beings, plants, fungi and some protistans. These kind of endomembrane system can be found in them. Right. We'll describe about what are these endomembrane system organelles. They include the 4 main organelles which I have told earlier. They include endoplasmic reticulum, followed by Golgi bodies, followed by lysosome, and finally the last one will be your vacuoles. Alright, these cell organelles include endoplasmic reticulum, Golgi body and lysosomes as well as vacuole. These 4 organelles are always coordinated to each other. First, everybody in the in the molecular biology, we know a concept of central dogma. Let me explain this entire concept to you with the reference of central dogma. We know about 3 processes. The DNA can replicate and can produce mRNA as well as proteins. The process by which DNA is replicating is called as replication. The process by which DNA is producing mRNA, it is called as transcription. The process by which from mRNA making proteins is called as translation. Now, carefully, clearly remember the site of the organelles or site of the region where this transcription and translation are happening. Right? First of all, transcription happens inside the nucleus, but whereas translation happens in a specific organelle which is popularly called as, any guesses? Which is popularly called as, yes, you are right, it is ribosomes. Popularly known as ribosomes. There is a region where it happens. Now, with this concept, let us try to understand the entire endomembrane system. Inside the nucleus, you have double helical DNA molecule. From the double helical DNA molecule, by a process called as transcription, you can produce mRNA. What is this? This is our so-called mRNA. Right. Now, this particular mRNA, what it has to happen? It has to get converted into the structures called as proteins. In order to get converted into proteins, it has to reach ribosomes. Now, in ribosomes, where are these present? Generally, in eukaryotic cells, these ribosomes are present either in cytoplasm. In cytoplasm, these ribosomes are present. Along with that, ribosomes are also present on the endoplasmic reticulum. Let me introduce to you the first organelle of our endomembrane system. That is none other than our endoplasmic reticulum. Right? Endoplasmic reticulum is the membranous extension of outer membrane of the nucleus. From the outer membrane of the nucleus, this endoplasmic reticulum will be formed. Endoplasmic reticulum is of 2 types. First one, rough endoplasmic reticulum. Second one, smooth endoplasmic reticulum. First one, rough endoplasmic reticulum means that the endoplasmic reticulum is looking rough when you are observing under the microscope. Now, how exactly is it looking rough? Because it is rough due to presence of these black granules. These are nothing but as the same as here. These are ribosomes. Endoplasmic reticulum has ribosomes on top of that. Now, an interesting point to remember, these ribosomes are attached to the endoplasmic reticulum by a small protein. This protein is called as ribofourin. By the use of this particular protein, ribosomes are attached to the endoplasmic reticulum, which makes the entire endoplasmic reticulum as rough endoplasmic reticulum. Now I told you that endoplasmic reticulum, or for convenience we can also call it as ER, it is of two types. One is rough, as you can see here, the other is smooth. Do you find any ribosomes on top of this? No. Now coming to the functionalities of endoplasmic reticulum. What happens? After mRNA being processed, mRNA will start to move out of this nuclear pore. You can see a small pore over here. Through that pore, this mRNA will move outside and it will finally reach our ribosomes. When it reaches the ribosomes, it produces a lot of proteins. That is a sequence, right? From mRNA, we have to produce proteins. Now, we're talking about these proteins. These proteins are formed inside these vesicles. Proteins are formed inside these vesicles. So, indirectly can I say, rough endoplasmic reticulum is the region for protein synthesis? Yes or no? Rough endoplasmic reticulum is a region where protein synthesis happens. Now what about smooth endoplasmic reticulum? Smooth endoplasmic reticulum does not have any ribosomes on top of it. Remember, rough endoplasmic reticulum will perform protein synthesis, and smooth endoplasmic reticulum, the so-called SER, it also produces some vesicles, but these vesicles are important for lipid synthesis and hormone. What are they? Lipid and hormone biosynthesis happens in smooth endoplasmic reticulum. Protein synthesis happens in rough endoplasmic reticulum. Now, both rough and smooth send their secretions in form of vesicles. You can find a small vesicle over here, which is emerging from the rough endoplasmic reticulum. Similarly, the smooth endoplasmic reticulum also releases a kind of vesicle also. This vesicle might even have lipids, or vesicles might even have proteins. Now coming to the second organelle, which is the part of endomembrane system. I have told you one thing, remember, there are several organelles inside this cell, but only 4 organelles are working coordinatedly. So the next organelle is immediately present beside it, which is our Golgi body. Golgi body, for the first time, was seen by chemical biology. The scientist has observed these densely strained particles, and the main function, write it down, main function of Golgi body is to, erase this, so it will be clear for you people to write it down. Protein packaging and protein modification. Along with that, in the previous year NEET exams, there were straightforward lines coming from this particular topic, that which is the major organelle responsible for glycolipid and glycoprotein synthesis. Don't be confused with the term glycolipid and glycoprotein. Everybody knows about protein and lipid. Add a sugar molecule on top of it. Protein with a sugar on it. Glycoprotein. Protein with a lipid on it. Glycolipid. This organelle is also responsible for glycolipid and glycoprotein synthesis. Right? Glycolipid and glycoprotein are synthesized here. Proteins are packaged, the so-called proteins which are coming from here, they will be packaged inside the Golgi body, and the lipids can also be modified inside the Golgi body. Now, Golgi body will also produce some secretions. From endoplasmic reticulum you have a vesicle. From Golgi body also you can clearly see a vesicle popping out. Can you see this, class? A vesicle is popping out. Now, there are two sides of Golgi body. One is facing the nuclear membrane, the other will be towards the plasma membrane. Other will be towards what? Other will be towards plasma membrane. Now, remember this. The side which is facing towards the nucleus, it is called as convex side. What is it? The side facing towards nucleus is called as convex side. The side facing towards plasma membrane is called as concave side. Convex side is also known as formative side or formative phase. Concave side is also known as maturation phase. Why the term formative and maturation? Because the vesicles are forming here and they are getting matured, because packaging, after completion of packaging, maturation of particles happen. Now, along with that, this this convex side is also known as cis phase and the concave side is known as trans phase. Now, you can find a vesicle coming out right here. This vesicle has three fates. This vesicle has three fates. Now, see, let's assume I am considering this particular vesicle which is having proteins inside it. It has entered into the Golgi body, and inside the Golgi body it has undergone some protein packaging and some protein modifications. After that, the same vesicle is coming out. So basically, technically, what I am meaning is this vesicle is also having proteins inside it. Yes or no? The same vesicle has been modified here and has been sent out. So typically, it will still have proteins only inside it. This particular vesicle can potentially be called as a lysosome. Yes, you are right. The third organelle is here. That is lysosome, which are popularly known as suicidal bags. I'll tell you the story why we'll call them as suicidal bags. But first of all, let us make sure that what are the three fates of this, secretory vesicle is. From the Golgi, the vesicle that is coming, it is called as secretory vesicle. First fate, it can be a lysosome. Option number 1. Second fate, if now consider this particular vesicle, it is also coming out of endoplasmic reticulum, but it is having lipids inside. It will also enter into this Golgi body. It will get modified inside the Golgi body, and it will come out and it will be delivered directly towards the plasma membrane. Now, why plasma membrane? Everybody knows it very well that plasma membrane is made up of lipids and proteins. These lipids will be delivered directly into the plasma membrane. Now, the third fate, the last and final fate of this secretory vesicle is none other than export. Now, some of the contents that have to be directly exported out, these vesicles, what happens? They'll go and bind to the plasma membrane, and after binding, they'll merge with the plasma membrane. They'll merge with the plasma membrane so that all their secretions are exported out. This particular process is called as exocytosis. What is it called as here? It is called as exocytosis. Right. We have completed everything about endoplasmic reticulum and Golgi body. We are left with two. That is our so-called lysosomes and our vacuole. Right. We were discussing about endoplasmic reticulum. We discussed about the types of endoplasmic reticulum, about rough endoplasmic reticulum and smooth endoplasmic reticulum. After that, the next organelle will be the Golgi complex, which has a convex phase and a concave side, a formative phase and a maturation phase, a cis phase and a trans phase, where we have clearly talked about which phase is towards the nuclear membrane and which phase is towards the plasma membrane. Now, we have talked about the phase of the secretory vesicle that will be popping out from the Golgi body. This has three fates. We have discussed about them earlier. It can either deliver its contents to the plasma membrane, or it can contain some proteins inside it, or it can directly send that particular structure outside of the cell through exocytosis. Now, the main thing that we are going to discuss is about when this vesicle is having or is containing proteins inside it. What are these? These are proteins. What now? If they are having proteins inside it. Biologically, remember one thing, there are two types of proteins. One are structural proteins, the other are enzymes. Enzymes are those structures inside our body which help in everything, literally everything. All the metabolic processes inside our body. Metabolism includes both catabolism and anabolism. Catabolism means breaking down of a complex molecule into simpler substances. And anabolism means from simpler substances you are constructing a big molecule. In both of these reactions you require a lot of enzymes. Now these proteins are made up, these enzymes are made up of proteins. That is what I want to say. Enzymes are made up of proteins. There is only one enzyme which is not made up of protein. Can anybody guess it? Yes, it is ribozyme. Ribozyme is the only enzyme which is not made up of protein. It is made up of RNA. The so-called RNA we are talking about here, right? That enzyme is made up of RNA, right? Coming to this particular state where the proteins which can be converted into enzymes. Now, this vesicle has budded off from the Golgi body. I am drawing it a little away for our convenience here. Now, understand the cycle here. The vesicle, the vesicle which is coming out from the Golgi body, if it is containing only enzymes, what is it containing? Only enzymes. The red color indicates enzymes, by the way. If it is containing only enzymes, it is called as a primary lysosome. It is called as what? Primary lysosome. Now what it does? Whenever you get infection by a bacteria or virus, these lysosomes will try to eat them. According to this lysosome, that particular matter, which is bacteria or virus or anything else, that particular matter is nothing but food for this. I am representing black color with food, right? When the enzyme is when the vacuole or this, not vacuole, secretory vesicle is containing only the enzymes. These enzymes are also not simple enzymes. These are acid hydrolases. What does this mean? Hydrolase, the meaning of the name hydrolase means by using water you are splitting molecules. That is the meaning of it. And at which presence it is happening? This particular process is happening in presence of acid. In presence of acid, if an enzyme is able to cleave molecule into its simpler forms, basically, if you want to understand in a simpler way other than being it very complex, the simple reason and the simple answer will be that these acid hydrolases will have the capability to break down carbohydrates, proteins, fats, nucleic acids, etc., entire cell. It can break down everything. That level of potent enzymes are present inside the lysosomes. Now let's try to understand the cycle of lysosomes. If the lysosome is having only enzymes inside it, then it is called as primary lysosome. When the same enzyme in the same lysosome, along with the enzymes it is having, if it is also having the food material. No, don't be confused when I say food material. Food material can be anything. It can be a bacteria or a virus or a carb or a protein. It can be anything. This is called as a secondary lysosome. Now what will the enzymes do? They will obviously digest the entire content, right? They will digest it and it forms to a tertiary lysosome. Now this lysosome is only having, what is it having? It is only having waste products inside it. What is it having, brother? It is having only waste products inside it. Now this tertiary lysosome will be sent out from the plasma membrane through exocytosis. I've told you everything, but I haven't cleared you why these lysosomes are called as suicidal bags. There is another lysosome which is quaternary lysosome. This quaternary lysosome is popularly known as autophagosome. What is it called as? Autophagosome. Auto means self, phagocytosis means breaking down, self-destruction, basically suicide. When this particular, the most potent one, when this vesicle having all of these enzymes, the quaternary one, when this enzyme or this vesicle breaks down, these enzymes have the capability to break down everything, ER, nucleus, Golgi, cytoplasmic contents, everything it can break down. That's why these quaternary lysosomes are called as autophagosomes. Is this clear? Now, we are left with only one cell organelle, and the most popular question which comes from vacuole is, what is the membrane of vacuole called as? The membrane of vacuole is called as tonoplast. It is only single membrane bounded, just like our lysosomes. It is also only single membrane bounded. Now, how can you imagine a vacuole? Vacuole is basically a dumpster. Cell pushes all of its contents into this particular vacuole, so that all the waste materials can be stored inside. Vacuoles can be of different types. There are some vacuoles which can store sap. There are some vacuoles, basically in some bacteria, in some Cyanophyceae members, you will find gas vacuoles. They are storing gas. Why? Reason? Yes, because of updrift. Have you heard the term buoyancy anywhere? Buoyancy means the ability to float. When you go to a water park or something, they give you a gas balloon. Why? Because it will maintain your floating nature. If the gas balloon is not there with you, you might sink, you might get completely sunk inside the pool. That's why they give you a gas balloon. The same property can also be shown by this vacuole. Mainly in amoeba and paramecium, you find a structure called as contractile vacuole. It helps in osmoregulation and excretion. Write down all the 3 points related to vacuole. And how is vacuole coordinated with this? From the start, if you see, we are finding transcription over here, translation, protein synthesis, protein packaging, modifications, etc. During all of these processes, all the waste materials that are coming outside will be dumped directly into the vacuole. Now, if you try to feel this entire concept, all these cell organelles are membrane bound and all of them are working coordinated to each other. Alright, let's discuss about some of the PYQs that we have observed in NEET and AIPMT and JIPMER from the previous years. Now, coming to the topic, in endomembrane system, a lot of questions are, repeatedly, they are coming from the same topic itself. Now, what can be these questions? The questions will be like, they'll give you some options and they'll ask you which particular organelle is double membrane bound. You can find nucleus is double membrane bound. People always get confused with this. People always go and mark only chloroplast and mitochondria as double membrane bound. But remember, you have to mark nucleus as well. The second one, rough endoplasmic reticulum is a site for which synthesis? Rough endoplasmic reticulum is a site for protein synthesis. Smooth endoplasmic reticulum is a site for which synthesis? Smooth endoplasmic reticulum is a site for lipid synthesis and hormone synthesis. Now, they'll ask you, which phase of Golgi body is towards nuclear membrane? You have to answer, is it convex, formative or cis? You have to mark any of this. When someone asks you which phase is towards plasma membrane, you have to answer concave, maturation or transphase. Right? And this is a popular question. What is a membrane of vacuole called as? What does a vacuole store? In some organelles, in some organisms, vacuoles store sap. In some organisms, vacuoles store gases inside them. They store ergoblastic substances. They store all the waste. In some, this vacuole can also be used as for excretion and osmoregulation. In which organism is vacuole used for excretion and osmoregulation? The answer will be amoeba, the main organelle. The main organism. Right? And they might ask you about lysosomes. Lysosomes contain enzymes. These enzymes are called as acid hydrolysis because they work at a pH of 4 to 5. They work at 4 to 5, which is acidic in nature. They contain enzymes to degrade everything. That's why lysosomes are also called as suicidal bags. Now, one small point that I have to add here. We're talking about, right now the current topic we're talking about an animal cell. Now, these Golgi bodies, these so-called Golgi bodies, inside plants, these are not called as Golgi bodies. These are called as, and start. The so-called Golgi bodies we were discussing in animal cells, these are not called as Golgi bodies in plant cells. Inside the plant cells, these are called as dictyosomes. And the so-called lysosomes which we are talking about, these are called as lysosomes only in animal cells. In plant cells, these lysosomes are called as spirosomes. And you will even get some questions that what are the organelles involved in endomembrane system. Answer starting with endoplasmic reticulum, Golgi bodies, lysosomes, and vacuole.